I welcome you all for today's session about enzymes. Today we are going to discuss an interesting topic on classification of enzymes. Before going into the topic, let me introduce myself to you. I am J.Mahalakshmi, Assistant Professor of Nutrition and Dietetics from the Standard Fireworks Rajaratnam College for Women. Enzymes. First we are going to discuss the meaning of enzymes. So enzymes are called biological catalyst. By catalyst, in a sense, it speeds up the reaction without involving in the actual reaction. Thereby enzymes are called biological catalysts. These enzymes are made up of protein, and these are highly specific, and particular enzyme acts only on the particular component. It won't interfere with the other chemical reactions, and these enzymes are reusable because it does not take part in any chemical reaction. So it can be used again and again in the human body and the biochemical reactions. For example, lactase is an enzyme used to break down lactose carbohydrate, which is a disaccharide, into glucose and galactose. So these lactase enzyme have high specificity, which acts only on the lactose component. And amylase enzyme, which breaks only the starch into simple sugars. So these enzymes which will not act upon any other substances, and while breaking down also the enzymes will remain unutilized, and we can the amylase enzyme will be used again and again to break down the other substances. So classification of enzymes, we have 6 groups of classification enzymes, 6 types of enzymes. One is transferases, oxidoreductases, hydrolases, lyases, ligases and isomerases. One by one we are going to discuss in this lesson. Oxidoreductases. The term oxidoreductases means both the enzymes which supports the oxidation reduction reactions. So the transfer of electrons from one molecule to the another molecule is supported by the oxidoreductases enzyme. As I said, these reactions involves either oxidation or reduction process during the chemical reaction. The examples for these enzymes are dehydrogenases and other oxidases kind of enzymes. So these oxidators enzymes are particularly used in the energy production mechanism, detoxification mechanism and antioxidant defenses mechanism. For example, in cellular respiration there are lots and lots of dehydrogenases and oxidases enzymes have been used to synthesize energy. And detoxification, for example, in alcohol metabolism, alcohol dehydrogenase enzymes are used to break down the alcohol component into the aldehyde component. So and antioxidant defenses mechanism, glutathione reductase is used to break down the glutathione to neutralize the free radical. So these are all about oxidoreductases, and examples are dehydrogenases, example alcohol dehydrogenase as we discussed, and oxidases example is cytochrome C oxidase. Cytochrome oxidases are enzymes present only in the liver, which will be used entirely to break down the toxic substances which get into the body during through food or medicines or any other toxic substances. Reductases like nitrate reductase and peroxidases like catalase. Peroxidases are enzymes which supports the oxidation processes, free radical processes. Okay, next we are moving into the second type of enzyme, transferases. Transferases catalyze the enzyme reactions involving the transfer of a particular group from one molecule to the another molecule. For example, in aminotransferases, in transaminase or kind of enzymes, the amino group is completely transferred from one amino acid to another amino acid. Thereby the essential from the essential amino acids our body synthesis non-essential amino acids. So certain type of certain types of reactions which involves transferases enzymes are by the transfer of methyl groups, by transfer of glycosyl groups, amino groups and phosphate groups. These chemical groups are transferred from one molecule to the another with the help of transferase enzyme. So for methyl groups, DNA methyl transferase enzyme is an example, and for glycosyl group, glycosyl transferase, and for amino acid group, aspartate transaminase. So there are many kind of transaminases and transferases group which converts the essential amino acid to non-essential amino acid in the human body. And phosphate group kinase. So in the phosphate group the phosphokinase, hexokinase kind of enzymes which transfers the phosphate group from one carbohydrate substances to the other. And then transferase play a very key important role in metabolic pathways, biosynthesis and all those. So, these transaminase reactions are involved in the amino acid synthesis. The amino acid synthesis in a sense that non-essential amino acids are being synthesized in the human body. So, metabolic pathways, glycolysis and glyconeogenesis and all these are involved in the energy metabolism. So, here is an example of hexokinase. In glucose, how glucose is converted into glucose 6 phosphate. The phosphate group is being attached to the is transferred from the adenosine triphosphate, and the phosphate group is being attached to the glucose 6 phosphate with the help of the enzyme hexokinase. And the second example is alpha ketoglutarate to L-glutamine. So, in pyruvate and L-glutamine with the help of alanine transaminase. So, from the alanine amino acid, along with the alpha ketoglutarate, the amino groups is being transferred from the alanine, and keto body like pyruvate is produced, and also the L-glutamate. And the third type of enzymes are hydrolases. These hydrolase enzymes involved in the cleavage of a molecule utilizing the water. So, by addition of water, the chemical component will get breakdown or will undergo the chemical changes. These reactions involve the breaking of peptide bonds. Example, proteases. When the protease enzymes are acting upon the protein component, inclusion of water will be there and peptide bonds are being broken. So, glycosylic bonds, for example amylase, ester bonds, for example lipase, and phosphoester bonds, example DNase. These hydrolases enzymes play a very important role in protein degeneration and turnover. So, as I said, the peptide bonds are broken down only with the help of the hydrolases enzymes. Protease, amylase and all these are hydrolases enzymes which utilizes the water molecule for the chemical reaction to occur. So, for a protein degeneration, protein breakdown, protein turnover and protein synthesis, everything requires the hydrolases enzymes. And carbohydrate metabolism requires hydrolases enzyme, and lipid metabolism, nucleic acid metabolism, all the metabolic processes requires hydrolases enzymes for breaking down the components. So, examples are protease, as we discussed, the trypsin and pepsin are the gastric enzymes which requires to break the protein substances at the peptide bonds, and resulting amino acids are eliminated or produced. And amylases, salivary amylase, pancreatic amylase, different amylase components are there in the digestive system, and lipases, it's pancreatic lipases, nucleases are all the components or all the enzymes which utilizes water in a chemical reaction. The example says A plus B in the when chemical reaction occurs, or H2O molecule, water is involved and resulting hydrogen bonds are added in the products, and OH group is added in the another product. Thereby the bond between A and B, either it can be a peptide bond or a glycosylic bond or any complex bond, that will be broken. And isomerases catalyze reactions involving the rearrangement of atoms within the molecules. So, for example, isomerases are enzymes which does not involve in any other chemical changes, but it only involves the structural changes. So, in the actually in the molecular formula of the chemical component will remain constant, but the structural formula will change because of the isomerase enzyme. For example, there are different types of isomeration reactions are there. One is structural isomeration, stereoisomeration and geometric isomeration. So, in all these the components molecular formula will be the same. For example, the glucose and fructose have the same molecular formula of C6H12 and O6, and in the amino acid have also the same molecular formula. But the structural formula will be different. In structural isomerization, glucose is converted into fructose, only the structure differs. Glucose contains the aldehyde group and the fructose contains the keto group. So, structural differentiation happens, structural changes happens. In stereoisomeration, the L amino acid is converted into D amino acids. It will be like a mirror image. The L and D amino acids are like a mirror image. So, that is stereoisomerism. Geometric isomeration, conversion of nucleic acids conversion of nucleic acids to fumaric acids. So, there is a geometrical, the structural changes will happen in the geometric isomerization. Isomerases play a key important role in again all the metabolic changes in the human body. The major metabolic reactions happens are carbohydrate metabolism, protein metabolism, fat metabolism and detoxification. These are all the metabolic reactions, and isomerases enzymes are involved in all the metabolic reactions. So for example, here I have given the glycolysis and glyconeogenesis flow chart. Here you will be able to see the involvement of isomerase enzymes. So the isomerase enzymes are required to convert the glucose 6-phosphate to fructose 6-phosphate. Here glucose and fructose are the same components, but with different structural formula. So with the help of isomerase enzyme only, the glucose 6-phosphate will be converted into fructose 6-phosphate. And here the 3 carbon compound glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are the 3 carbon compounds which can be interchanged its structure with the help of isomerase enzymes. So both have the same molecular formula, only structural formula will change, structural structure of the component will change. That by the help of isomerase enzymes, these components can be changed from one to another. And the next group of enzymes are ligases. Ligases catalyze the reaction involving the formation of new chemical bond between the two molecules. So ligases are actually the attaching molecules. Attaching molecules means two different substances are, for example, hydrogen and oxygen are there. The molecule H2O, water, if you want to produce the water, we need certain connection and chemical bond between the component. So such chemical attachment happens in the human body with the help of ligases enzymes. So these involves peptide bond formation, glycolytic bond formation and carbon-carbon bond formation are all these examples. For example, tRNA synthetase enzyme, with the help of tRNA synthetase enzyme, the peptide bonds are synthesized. The tRNA is transfer RNA which is required for coding the protein. So with the help of the enzyme, the protein will be coded and peptide bonds will be synthesized. So similarly, the examples are there for glycosidic bond and carbon-carbon bond also. So and ligases play important role in protein synthesis. The protein synthesis, for example, in case of protein synthesis, our body, we consume proteins in the form of protein, different kinds of protein, from animal protein, from plant protein and all those. So when the protein get digested and absorbed in the body, it will be in the form of amino acids. So from the amino acid, different kinds of body proteins are synthesized in the human body with the help of the genetic material, with the help of the codes produced by the genetic material like DNA and RNA. These RNA synthesis the protein like messenger RNA, tRNA, these genetic material will code the proteins for synthesis. For example, any kind of hormones, enzymes, structural proteins like cartilage, collagen, elastin, keratin, all these proteins are synthesized in the body. So all requires the ligases enzymes for synthesis. And DNA replication and repair. So DNA replication, during replication also DNA ligases are involved to replicate the DNA. Cell wall biosynthesis and metabolic pathway regulations. All the metabolic pathway regulations we required enzyme synthesis, hormone synthesis and all those. For regulation, these ligases are used. So these are certain examples for ligases. In plasmid, the plasmids are attached to the foreign, this is a recombinant DNA technology. So by this the plasmids, the DNA from another substances, another component is, another living cell is being isolated and particular DNA is being attached to the plasmid with the help of the DNA ligases enzyme. So and the last category is lyases. So these catalyze the reactions, involves a cleavage of chemical bond without hydrolysis or oxidation. So the breaking of bonds happens here, but without oxidation or hydrolysis. So here the reactions are carbon-carbon bond cleavage. The cleavage, the breakdown happens at the carbon-carbon bond and carbon-nitrogen bond and carbon-oxygen bond and carbon-sulfur bonds. So all these places the cleavages means the separation happens. The enzymes involved are aldolase, arginase, decarboxylase. These are all the enzymes involved. Aldolase involves the removal of H2O molecule from the chemical reaction, and by decarboxylation removes the carbon dioxide component from the chemical reaction. So, and lyses are involved in the detoxification, cell wall biosynthesis, amino acid metabolism and all the other metabolic pathways. For example, decarboxylation reactions happens in many metabolic pathways. So, all these are with the help of lyase enzymes. So, this is a Krebs cycle. In these the decarboxylation reactions happens between the oxalosuccinate to alpha ketoglutarate and alpha ketoglutarate to succinyl CoA. So, here in both the reactions the decarboxylation reaction happens. So, finally conclusion. So, these enzymes plays a very important role in the biochemical mechanism that happens in the human body, either in the terms of metabolism, detoxification, waste product synthesis, urea and creatinine synthesis, DNA replication, cell wall synthesis, and all these biological processes are carried out by the help of enzymes. So, enzymes are very crucial for life. So, if a body is lacking a particular enzyme, the person will enter into a stage, pathological stages the person will enter into. For example, if a person is lacking lactase, the person will end up in lactose intolerance. So, these enzymes are highly specific and efficient. Highly specific, the particular enzyme will act only upon the particular component, and those enzymes which will not interfere with any other chemical reaction. So, isomerases will change only the glucose into fructose, no other enzymes it will go and affect, no other chemical reactions it will go and interfere. So, these 6 main enzyme classification have distinct functions and maintains the biological system and supports the human survival. These are the references, you can also refer, and thanks for watching this video. We will meet in the next session. Thank you.